From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 34, recorded on September 26, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Morning. How are you all?
Pretty well. It looks like you're wearing a flannel shirt. Is it cold out there or something?
Yeah, we finally got that nice fall weather. It's beautiful right now. Low 70s. House is actually, I think this morning was 45 this morning.
Wow. Also joining us from here in New York, Tim Chung.
Hi, everyone. Yes, it's also very nice in New York right now.
Yeah, nice weather.
Lovely to be outside.
And from New Orleans, Vivian Morrison.
Hi, guys.
Hey, Vivian, the students down there invited me to do their retreat next year.
Really?
Yeah.
Explain.
Yeah.
Cool.
And so while I'm there, I'll say hello to you.
Yeah, please do.
I think it's a Thursday and a Friday in April, end of April.
Is it with the Department of Tropical Medicine and Infectious Disease?
I don't know. I thought it was microbiology and immunology. But
it could be. Well, I mean, I imagine they're on the downtown campus as well.
Downtown campus.
Why are you laughing?
Because that's what we have. We have the medical school, and then we have downtown Columbia.
Downtown is the medical school in this case. But yeah,
downtown Columbia is still pretty uptown.
Yeah, it is. But
it's still a city.
It's, with reference to the medical center, 168th and downtown is 116th. And you and I are even further downtown, Tim, yes?
Yeah, I'm down in the equator.
You're 30th Street, right? More or less?
Yeah, 30th Street, exactly.
And that's exactly where I am, except I'm on the west side and you're on the east side. So a little bit of New York geography there. But speaking of geography, we have some science to tell you about today, and the honor falls on Vivian. So take it away.
Yeah, thanks, guys. So the paper we're going to be discussing today the title is Microglia Coordinate Cellular Interactions During Spinal Cord Repair in Mice. And I guess I'll do what Ori does, which is say that the first author is Faith H. Brennan, and the senior author is Philip G. Popovich. This is if you were to pull up the paper and look at it, you might initially, there are probably two things you would think. One is, man, they've got a lot of figures, and it's all, looks like art rather than science.
It's very pretty.
It is very pretty.
And it's open access, so you can just download it and then hang it on your wall.
Mm-hmm. And there are a couple things that, yes, you could. They're very, very pretty. But there are a couple of things that, that I wanted to talk about today that would help people who are listening, who want to go find the paper on later on, that would help them know how to approach it. So as you can see in the title, we're going to be talking about one of the glial cell types in the brain, microglia. And this is in contrast to macroglia, which contains astrocytes and oligodendrocytes. And microglia make up 10% of the brain's cells. And so pretty small, but they're really, really important and for many things. And I would say that the microglial field, probably more so than any other field, is just booming. Especially with the technique that's used in this paper, which is single cell RNA sequencing. So another thing that's interesting to mention about microglia is, in contrast to macroglia, is that microglia are not born in the central nervous system, whereas oligodendrocytes and astrocytes are. The microglia are born from multiple places outside the CNS at multiple times in an organism's development. And there are, like I said, this field is blossoming, and one aspect of that is understanding how these different populations of microglia that arrive at different times and their outside the nervous system correlates, macrophages and monocytes, how those are all related. And then to add a couple dimensions onto that, you can talk about how they may differ spatially with respect to the nervous systems of different brain regions or the spinal cord. You can talk about how these populations might vary with age, and then how they're going to be differentially impacted during disease or injury. So it's just a really fascinating field to be a part of, and I'm carving out a little place for myself in it as well.
So a quick question, Vivian.
Yeah.
So neurons after a certain age, you no longer have, by and large, don't have new neurons born, except for like adult neurogenesis.
Depends in humans.
In humans,
in mice, it's definitely more robust.
So for microglia, so they come from outside the central nervous system. Do they eventually stop going into the nervous system at some point?
Yeah, their arrival in the central nervous system, and when that period of invasion ends, depends on the development of the blood-brain barrier, which we may have talked about before. And the blood-brain barrier is something that's kind of built by the cooperation of endothelial cells and other perivascular cells. So the endothelial cells are the cells that actually make up the blood vessels, as well as astrocytes, which use these specialized processes to wrap around the blood vessels. And they basically are going to regulate what goes in and out. And so in the case of disease, where you're going to have peripheral immune cells coming in to help, there have to be changes at the blood-brain barrier to allow those cells to enter, which is actually something that they're going to talk about here. But yes, in development, the blood-brain barrier and its closure is what's going to regulate the entrance of microglia into the central nervous system. And as we age and in disease, the integrity of that barrier changes and goes down, and makes it easier for peripheral cells to get in. And so that's one of the things that people are looking at, is trying to understand in what situations do we have peripheral counterparts, so monocytes, coming into the central nervous system and contributing to like age-related cognitive decline or neuroinflammation more broadly.
But microglia are really proliferative, right? So there's been experiments where if you ablate almost 99% of them in the adult brain, they can restore themselves very quickly within a couple of, like a month, I think. Yes,
that is true. And so what's interesting about that is in the resting brain, the microglia are not very proliferative. So the proliferative response is one that shows up during injury and disease, infection, that kind of group of things. So I just totally lost my train of thought.
But yeah, we see in this paper as well. That
was I was going to bring up.
It goes up like crazy in this paper.
Mm-hmm, it does. So one of the things that we do in science is, if you want to know how something works, you have to stop it from happening and see what happens. So like if we want to know what a gene does, we would knock that gene out. And then the next step would be, well, let's make more of that gene. So they're going to do both of those things here. So they use a drug that depletes microglia. It inhibits a receptor that would bind to a growth factor that's necessary for survival of microglia. But I will also note that it's not specific to microglia. It's also going to impact peripheral monocytes, which I actually see as being an issue in this paper that is not addressed. They don't talk about how. I mean, they don't talk about this. They do not discuss this. But so Jason mentioned that the microglia can repopulate. They will repopulate, and pretty quickly. But they're not going to do it as quickly as the peripheral, like bone marrow derived monocytes are going to. So oftentimes when one is using these pharmacological methods to deplete microglia, you have a short window in which to kind of catch the brain in a microglia deficient state without the caveat that you've also depleted peripheral macrophages and monocytes, which could have impacts on the immune system and interact with the CNS in that way. But yes, that's a very good point that I was going to that I lost there for a second. So I think that I didn't really mention what microglia are, though you probably could have guessed it has something to do with the immune system because they are derived from macrophages, at least some of them. And macrophages are like the microglia and macrophages are very similar. I like to think of them as garbage men, but also first responders because they are scooting around the body if you're a macrophage, or the brain and spinal cord if you're a microglial cell. And they are engulfing material. They're engulfing dead cells. They're engulfing or degrading extracellular matrix. They're engulfing synapses. And in doing so, it puts them in a great position to kind of see what's out there. And so they might, for example, phagocytose or eat a piece of a cell that's been infected by some pathogen, and then it has access to parts of this pathogen, which it can then present on its surface and in that way start interacting with immune cells and saying, hey, we need help here. Let's recruit peripheral immune cells to come into the CNS and help manage this infection. So they're garbage men, but they're also the ones who are, like I said, first responders. And they're also going to be large producers of inflammatory cytokines, but also anti-inflammatory cytokines. And so a theme of this paper is that microglial balance is key. The balance between inflammatory pathways and anti-inflammatory pathways is really, really important. This is true in development, and it's true in the aging brain and in disease. And so it's really just saying the same thing that a lot of people are starting to say, with the added benefit that it's looking at a specific interaction between microglia and the balance of what they produce. And that the interaction of that with peripheral cells in a particular context, which is spinal cord injury. So like a contusion, I think by and large, when they are saying SCI, spinal cord injury, they're talking about a contusion injury, which is much more relevant clinically than some of the other methods where you actually go into the spinal cord and you take a pair of tweezers and you smush it. That doesn't happen to humans. So, and that's going to have a different impact than one in which the spinal cord is intact by its extracellular environment, which is what was going to happen in humans when they.
So I actually had to look up what contusion is, and it is.
Oh, contusion is like like blunt force trauma, like a bruise, like.
Yeah, it's like bruising, but then I can't really picture what a bruise on a spinal cord would look like. But I guess
there's
it might actually look like a
blood.
Bruise. Yeah, there would just be blood going in.
There is. Because not only do you have like these, these molecular or chemically mediated openings and closings of the blood-brain barrier, you also have frank destruction of blood vessels. So there is blood there, and one of the things that we find that they bring up as being important in these microglia and how microglia normally function in spinal cord injury is by processing iron, which can be very reactive, and so it has to be picked up and cleared. And so, like I said, we were talking about the balance of inflammatory and anti-inflammatory. People tend to think about inflammatory as being bad and anti-inflammatory as being good, but it's just really not that simple. It's not black and white. So little in life and in biology is black and white. And so if someone is to go and look up this paper, I think you need to, or any other paper related to a similar topic, you need to keep in mind that there is no like good or bad necessarily. There's, there's pro-recovery outcomes. There are things that are going to delay recovery or steer the response in one direction, but it's just really hard to use that dichotomy, good and bad, to describe microglia. And so that's something that the field is trying to break away from. Traditionally, they've even used like activated versus homeostatic, or resting actually is the word that they used. And they've also called it M1 or pro-inflammatory and M2 anti-inflammatory. And, people are just saying now that it's just really not that simple. So I would say try to break away from that kind of thinking.
But another digression. Sorry, but I'm just wondering.
No worries.
Like for virus, like if you're infected with a virus or a bacteria, it makes sense that you want to get some inflammation going to fight off that infection. But if you have a bruise where you might be someone hit you or you fell over yourself, and some injuries inside your body, but you're not infected, does inflammation actually help, or does it actually hinder? And it just makes you suffer from pain, such that if you were to give someone who had a bruise some anti-inflammatory, like an ibuprofen, does that make it worse, or does that help? Do you know?
The inflammation,
the recovery, like if you block the inflammation.
If you were to give, well, so the inflammation is necessary. So when you have a bruise, and especially in the central nervous system, there's going to be a lot of stuff that's dying and degrading. And so, okay, let's talk about cell death for a second.
Sorry about that.
No. I think it's an important question because, we think about the immune system, and it's like you either think about it as being like the savior in a situation where there is a pathogen that needs to be taken down, or we think about it as being a really bad thing when we think about like cytokine storms and rheumatoid arthritis and lupus and stuff. And certainly, it's like with in medicine, we say any medicine can become a poison. It's the same thing with microglia. You need them to be inflammatory to a point, and then there are lots of systems in place that we're trying to understand to control, that are there to control the inflammation. But so cell death, like we have necrosis, which is an unregulated dying of cells. And then there's apoptosis, and there's probably other tosises. Everybody likes to have their own word for like the very specific kind of cell death. But in general, necrosis is unregulated. So you just have like straight up damage to the tissue, and cells are exploding and spewing their innards into places that don't normally see that. And then you have apoptosis, which is a regulated cell death. And so there are clearly defined steps that happen, and the cell, which was once whole, breaks up into little bubbles that contain the innards, and those can be like easily cleared by microglia. And if you don't have inflammation, if you don't have any microglia or invading, blood-derived monocytes, if you don't have any sign that says like something is wrong and I need help to clear all of this apoptotic debris, then there's an idea that apoptotic debris, which has on its outside signals that are specific to apoptosis and say, you need to come eat me, come find me and eat me, that those signals over time are going to degrade and disappear. In cells, they have all this machinery inside that contributes to turnover of protein and lipids and basically, keeping things healthy. But these apoptotic bodies, the debris, they don't have organelles. So they don't have the option of, switching out proteins or lipids on the outside. So that stuff ostensibly is going to disappear, and then what was once apoptotic is going to become kind of necrotic, and then that's going to lead to like more inflammation because you have like all of this stuff that's not normally seen by the immune cells. So it's kind of like a self not self situation.
So like if a garbage man doesn't come and clean up the mess, it's going to rot.
Yeah, and you get cockroaches and rats and all that sort of stuff.
Yeah.
But you need
in New York, we know about this.
Yes, in New Orleans too. Like I just found we were sitting at the park yesterday, and I picked up my bag, and there was just like this massive cockroach that had like been inside of it.
Yeah.
But anyway, I'm like developing a tolerance to it. So it's good. It's like exposure therapy. But so you need the inflammation to bring in enough cells to manage clearing apoptotic cells and necrotic material before it starts leading to cell death, like neuronal cell death. Because it's not just like proteins. There's also all of these molecules like neurotransmitters, glutamate or calcium, ions that are could be excitotoxic for neurons. And then you would just have like a increased degeneration, secondary degeneration. It's not actually caused by the initial injury. It's rather caused by the fact that initial inflammatory response is not there.
Okay.
Additionally, and I think this would be an interesting discussion, but I'm not sure what the answer is. I suspect that without the initial inflammatory response, you may not be able to activate anti-inflammatory pathways. So you may actually have a prolongation of the injury. And then what kind of long-term impacts that could have on function, that's up. That's another discussion that would probably be very interesting. So as I mentioned previously, like usually our first thing, the scientist's first step is to try to knock out or block the thing that you want to study from happening and seeing what happens. So, and as we were talking about earlier, you can pharmacologically deplete the microglial population using this drug, which I'm just going to call PLX, but that's the inhibitor of this growth factor receptor. It's necessary for microglial survival. So they see that, so they'll knock out the microglia for a certain period of time. And then they perform the spinal cord injury. And then at different days, they'll look at different time points, really just 2 time points over the course of recovery. And they'll look at those time points in terms of histology. So they'll look at staining of tissue, labeling of different cells and proteins. And then they also they mention this very briefly, but it's important. They do some behavioral and functional tests because, as I have just recently learned in the context of my work, which is now starting to involve stroke, the histopathology doesn't always correlate one-to-one with the function. So you might have like a lesion that looks less inflammatory or like not as large, but it may have the same functional outcome as a larger lesion. So there's definitely, it's a little bit of a black box. So they use PLX to knock out the microglia. They perform this spinal cord injury, and then they perform these behavioral and functional assays. And they find that 30% of the mice that only 30% of the mice that had their microglia depleted actually achieved the same level of performance, same level as recovery of the animals that were treated with a vehicle. So the animals that still had microglia. And I think that, you're like 30%, like it's a lot. Well, as we were talking about, a lot of our interventions are not sealed up tight. They're not bulletproof. We can't eliminate all the cells and still be able, we can't have our cake and eat it too. We can't eliminate all the cells, but then have a period of time or a window in which we can do our experiments that are going to rule out certain caveats. So I suspect that the fact that they don't have, it's that we have 30% of animals reaching comparable performance as those that were treated with vehicles is due to that. But they show that these behavioral, this poor performance on the behavioral assays is associated with histopathology. So infiltration of, there are a couple of things that they look at. So they find that there's axonal injury, and they see that there's myelin degeneration. So myelin is the coating around axons that helps insulate the signals that constitute neurotransmission. So then, as I may or may not have mentioned, the microglia, they actually don't work alone. They're going to, in spinal cord injury, they're going to recruit peripheral immune cells. So they're going to recruit circulating monocytes and bring them into the tissue. And those cells are they're hardcore phagocytes. So phagocyte means eating cells. So they're more prolific than microglia. So their job is to go to the lesion's core and really, soak up, sweep up, degrade all of that debris that could be inflammatory if it's not cleared. And we also want that stuff to be cleared because we want to try to make an environment in which regeneration could potentially happen. So you've got to clear out the debris. And around that core we see a development of something called the glial scar, which we've probably talked about, but it's a kind of fibrotic cocoon that the lesion is surrounded in, and it's generated by astrocytes and NG2 cells, which is what they call them here. And I will just say that they're fibroblasts-like. I think people are not really sure what they are, where they come from. There's definitely some something that's unclear there, but most people focus on astrocytes because I think they're probably the larger component of the glial scar. And so the idea of the glial scar is to kind of cordon off this lesion, prevent inflammatory cells, inflammatory material from entering normal appearing or normal tissue, uninjured tissue.
The pictures are great. I've never seen a glial scar before, and it does look like a barrier surrounding the injured site. Yeah, it's
really cool. And it's a lot easier to see these kinds of things in the spinal cord than in the brain.
Yeah, in the brain you just get gliosis, which is just like a clump of more astrocytes, I guess. Yeah.
And also the spinal cord is simpler in its axes, and the way it's laid out. It's just a tube, and it's got multiple tubes on the inside, but, with the brain, you've got stuff kind of going in a bunch of different directions. And so it's kind of hard to visualize it unless you have a brain that has been cleared where all of the lipids have been removed, and then you do fancy imaging.
We talked about that last time.
Mm-hmm. Oh, did we? I can't remember. Yeah. It always feels like an eternity between different episodes. So I mentioned that the microglia and the monocytes that have been recruited, so they become macrophages, they work together to help deal with all this stuff resulting from the lesion. So what they saw is when they depleted microglia with this drug, PLX, that the macrophages were not localizing to the core. And in fact, they were migrating into normal appearing white matter. So they're actually, let's not talk about their function, but they're in places that they're not supposed to be. And so you can already see the potential for this bystander injury, like stray bullets, basically. And they mention also that these macrophages are foamy, which means they have lots of vacuoles, which is usually an indication that there's an increase in their intake, so phagocytosis, and they have that's also associated with a more inflammatory macrophage phenotype. So they, in addition to this kind of problem with recruitment and localization of the macrophages, they also see that the glial scar is attenuated. So the microglial presence is in some way, from this, they conclude that the microglial presence is in some way regulating like the movements of the macrophages and in some way influencing astrocytes, since the astrocytes are the major contributors of the glial scar. So they also confirm that the astrocytes are impacted because by showing that it's not just that like the proteins that are involved in making the glial scar are decreased, which is one thing that can happen when you're staining or labeling a particular protein. You might not see it just because that one protein is no longer part of the scar, for example. But just like with microglia, they become proliferative after they're like activated. Astrocytes do the same thing, and they have to proliferate to generate a large enough population to make this like nice sealed up scar. So they look at proliferation of astrocytes, and they show also that the proliferation is reduced. So there are programs that are involved in the reactivity of astrocytes to spinal cord injury. They're not coming online when microglia are not present. So they show that these findings are pretty much replicated by 2 other spinal cord injury models. So we're really looking at something that's some relationships, intercellular relationships that are conserved across different kinds of injuries. So now they move out of the tissue because we want to get a better idea of like a little bit more resolution and understanding what's happening in the tissue. So they're going to do bulk RNA-seq. This is different than what I mentioned earlier, single cell RNA-seq. Bulk RNA-seq is where you just take like a big chunk of the spinal cord and you dissociate it into a single cell suspension and you isolate the RNA from everything. And you can just see what genes go up relative to whatever your control is. So it could be, in one case, they're going to compare spinal cord injury versus sham animal. So the sham would be like, in the case of the crush injury, it would be an animal where the spinal cord is exposed but not crushed by the forceps and then closed again, which of course is going to generate its own, it's an injury still. So that's why it's important to have like the animal that didn't get the crush but mostly got everything else.
Oh, wow. And I didn't realize they have to like open up the bones and stuff.
I mean, I've never done it, so I don't know to what extent that happens. But for example, the lab that I'm working in does a lot of stroke models. So they do a middle cerebral artery occlusion, and they have to open up I think they go in through the carotid. So they have to open it, and they just like poke the carotid artery. And just the poking actually causes the blood vessel to go into spasm. So you want to know that like that's going to happen in both conditions. And so any difference that you see between the 2 conditions is probably not due to the vascular spasm. So that's why having those kind of procedural controls is so important.
This is this bulk RNA-seq is powerful, but you can look at this figure; it's beautifully done, but it all comes down to this idea of gene ontology to say what are the general things that are going up and down. And gene ontology is based on, sort of weak categorization of each gene being involved in X process or associated with something. And this is where I really sort of the field over relies on these gene ontologies to say, oh, look, this is the thing that's going wrong in this process because there's a bunch of genes that are clumped together in this thing. But when you delve into the actual genes and their function, it's, maybe one paper that somewhat describes what the gene does. And so, basically you get a list of genes and you can make your own story is what I get out of this.
Yeah. So I don't know if anybody else wants to comment on that, but I will say, since part of my work right now involves going through some bulk RNA-seq stuff, and, this is applicable to really any RNA sequencing. there's a lot of redundancy. Like you can do a Go analysis, that gene ontology analysis, and it'll pop out like 18 pathways or phenomena, and they're like all kind of the same. And so you're like, well, so how do they differ? If it was different enough that it needed to have its own category, like what is that difference? And it might come down to like group A or Go pathway 1 and Go pathway 2 differ by only a couple of genes. And so what impact do those genes have that they could cause this, this one process becoming 2? And so what I think is admirable about this paper is that toward the end, they do try to isolate a couple of things, just, to be like, this receptor and this ligand, like somehow their relationship seems to be important for the phenomena that we're seeing.
Right.
So I think while it is by no means as deep an analysis as it needs to be for us to really understand what's happening, at least this paper shows that there are different levels of analysis that need to be done. Bulk has its role. Single cell has its role. All of those things need to be validated through other methods, even ones that are like much simpler, like immunolabeling, the staining of the tissue. And that, there's this whole other forest of information that we can get, but it's going to have to happen through you're going to have to choose to focus on something. And I'm sorry, like that's just how it's going to happen. You can't like figure out the unifying theory of everything by looking at this
you obviously
scale.
have to validate some of the genes that you want to follow up. But that's what I mean by sort of choose your own story, where it gives you a roadmap, I guess, to say, okay, well, maybe we need to look at this particular process. So that's fine. I think it's just in the field right now, there's data dumps where, papers are just saying,
that's my problem.
Things go up, things go down. We think this is what's going wrong.
Yeah. And, part of the reason that I'm just like, hey, look at the paper and just look how pretty the figures are is because you don't actually need to like go look at them very deeply. I mean, I think the figures are there for people like us, perhaps, who are looking for some sort of catch, who are looking for a lead that they want to delve into. But by and large, the findings of like all of those figures can be summarized into like sentences where, like, for example, the main effect of spinal cord injury is that a lot of genes go up. And the main effect of spinal cord plus PLX, so microglial depletion, is that there are a lot of genes that go down. But, interestingly, it's not that the increase in downregulated genes is not due to the fact that you are missing a population of cells, which I think is actually very interesting because it shows you, or it suggests that those microglia, like, yeah, sure, those genes are missing because the microglia are not there, but their presence is actually having network-wide impact on transcription and like the identity and function of other cells. And that by and large, the microglial presence is trying to upregulate certain pathways, activate certain pathways in the cells that it's recruiting, so the macrophages, or in the cells that are nearby, the astrocytes. So like, I have a bunch of notes here that essentially, boil down to the fact that, there are issues. They basically, like, maybe it's a little bit of choose your own story. And I think we'll only know this if one of us took time, or somebody took time to go into the data, because these papers do make or sometimes make their data available. Of course, it's in a form that's like impossible to consume unless you like know exactly what you're doing. Like, I look at it and I'm like, I'm not touching that because I have no idea what to do with it. But we won't, so we can't tell. Most people are not going to be able to tell if it's a choose the story that, or pick out the pieces that you want to use to support your story. But they show changes in gene pathways and networks that are associated with the changes they see in the poor recovery and the myelin degeneration, the foamy macrophages, the fact that the macrophages are not in the lesion. And so like just to bring up a couple of ideas or a couple of things that they show that like lipid processing and phagocytosis and iron processing go down. They show that molecules involved in chemotaxis, so that is a cell's migration in response to the release of some factor from like a dying cell, for example, or a microglia secreting these chemotactic signals that are recruiting and saying, hey, come on in to the macrophages. Those things are going down. Adhesion molecules that would allow the macrophages to get into the lesion, those are going down. So there's a whole bunch of things going down because the microglia are not present. And I'm trying to think if there's anything else to mention on that point. But I mean, you read the text and you're going to see like this thing goes up and this thing goes down. Oh, and additionally, if you have spinal cord injury, this thing goes up, and when you deplete the microglia, it just fails to go up. It's not that it's down-regulated; it just fails to respond. And then add on top of that the fact that you can look at 7 days post-injury and 28 days post-injury, and you're going to see differences there too. Like the population, when they do their single-cell RNA-seq, they're going to show like, at baseline, this is the composition of the microglial population. At 7 days post-injury, it's going to shift like this. But then at 28 days, it's going to shift like this. And it is a little bit of a data dump. They're like, there's this, these waves are happening, but we can't really necessarily get to, or it's not the goal of this paper to tweeze it all apart. But in terms of the manipulations they do, they try to provide some more, I think, potentially clinically relevant information, or just some more resolution and granularity to what's happening. They find that there are kind of two nodes that normally go up, or that go up after spinal cord injury. So in just baselines, like uninjured sham animal versus injured animal. And those two nodes involve these genes called CCL2 and TLR2. So the CCLs and CCRs and CXCRs, those are chemokine receptors and ligands. And then TLRs are toll-like receptors, and they're pattern recognition receptors. You have different versions of them placed in different parts of the cell to recognize different pathogens or components. And so they see that those two nodes fail to rise when microglia are not present in the injured spinal cord. So they say, this is kind of like the second thing that the scientist does is like, well, can we rescue something? Or what if we put more of this thing in? What do we see? And so they have their PLX-treated animals, they perform the injury, and then they inject CCL2 and an agonist or an activator of the TLR2 receptor. And if you had poor outcomes in your PLX-treated condition, where there's this failure of the CCL2 and TLR downstream pathways to be activated, when you add those back in exogenously, you see better performance. So they were able to, even in the absence of microglia, they were able to improve the animal's recovery just by stimulating or activating these the pathways that are downstream of CCL2 and TLR2. But as I've been saying multiple times, balance is key, because they found that if you introduce those two components, that agonist and the CCL2 protein, and the microglia are still present, and this kind of makes sense actually, you actually have worse outcomes on recovery. You just have too much. So microglial balance is key. And we all want to know, what are the pathways that controlled microglial balance. It's what I'm working on, and it's what many other people are trying to understand. And it's just really complicated by the fact that every injury model is going to be different in large or small ways, and the interactions between those differences and age and developmental stage and the organism, the nature and nurture that an organism has seen, all of that's going to interact. So, I should just throw my hands up in the air and say, like, we're never going to know anything, But
this, I kind of like this finding that when you get rid of the microglia and you have a spinal cord injury, you don't have these CCL2 and TLR2, so you have a really poor outcome. You kind of supplement them and you get better. Mm-hmm. But in a normal mouse where you do have a microglia, if you just dump a bunch of CCL2 and TLR2, they get actually much, much worse. Mm-hmm. Like the amount of macrophage in the spinal cord injury side is like crazy. Mm-hmm. And then it just reminds me of like when I listen to the immune podcast and the immunologists talk about things, they always go on and on about this Goldilocks kind of finding in immunology where something has to be just right. If you have too much of something, you get worse. If you have too little of something, you get worse, and it has to be just right.
Yeah.
Yeah.
Does anybody know? So like I said, this could be clinically relevant, but there is, I think, never a time when a human is going to be like a human who's suffering from spinal cord injury or whatever, is going to be devoid of microglia. Like they're always there. So like
unless they take the medicine that depletes microglia, which apparently is used for certain cancer.
Oh, okay. So really interesting.
Yeah, I kind of looked
at
it up.
Like that also it leads to side effects like brain fog and whatnot. We don't know what. I mean, some of those drugs are pretty nasty in terms of side effects.
Mm-hmm. Yeah, but that was exactly what I was getting at. Does anybody know like if that can be administered to humans and what the effect is? But it's like if the disease doesn't kill you, the medicine will.
I mean, you'd love to have a peripheral only blockage or some sort.
Mm-hmm. And also, I mean, if you're, it means the person's going to be severely immunocompromised.
Yeah.
And probably also, opened up to autoimmune disorders. Episodes. I would imagine. So we talked about the histology and behavioral stuff and the bulk RNA-seq, and then they take it a little bit further and they do single-cell RNA-seq. And the reason that one would want to do single-cell RNA-seq is so that you can see if there are different populations of microglia. Like, they were talking about the percentage of cells that are, focused on processing lipids goes up at this time. You can only know that if you use single-cell RNA sequencing. So they're looking at cell type specific or cell subgroup specific changes. And I don't really need to go into much there since we've already talked about it, and you can just read the text. It's extremely simple because it's all just up and down.
But this is the one where actually I'll just quickly interject. If listeners are interested in single-cell RNA-seq, TWiN actually did an episode on it eons ago, TWiN 11, $1 per neuron, where they talk about how to sequence individual neurons using these fancy barcoded droplets.
Exactly.
And the second thing is that these single-cell RNA-seq graphs are the one that has amazingly pretty pictures and colors together.
And that's what I'm going to talk about, actually. That was one of the things, like if we don't have to spend time talking about the bulk of the, no pun intended, RNA-seq results, we should talk about how do you look at these data and what kinds of things can you actually say. But I will be brief, ish. And there are two links that I think that we will add into show notes if those exist, because one of them talks about something that they do, but that I think is really cool, but they don't really talk about it much, and that's they use a statistical method to generate kind of a fourth dimension to their RNA-seq analyses, and that is adding in time. So they create something called pseudo-time. And what's really cool about it is that it allows you to sketch out like the developmental trajectory of a cell population or how it evolves over the course of, say, a spinal cord injury and recovery to that. And so, like, for example, if I was to put it into like a non-cell biology relevant example, it might be like where we have lots of canals here in New Orleans. So if you have a canal, it's just this one stream and it's all heading in the same direction. But little by little, we have, streams that are diverted and they'll go to different locations. And those pipes that they're taking, they go to different destinations. They, might have more or less lead in them, or they're larger in diameter or smaller, or they're more turbulent. And all of those things are going to change, what happens ultimately to the water, like how long it takes to get from place A to place B. And like, if it has more lead in it, for example, at the end. So these developmental trajectories are interesting. And I think it's cool that I'm seeing that more and more. And so I have a link that I think is cool to look at and can kind of explain.
It's really cool that they can just do it computationally and try to pull out where a cell is in its developmental trajectory or whatever.
Mm-hmm. And I think, but there's also something like a little caveat is that the reason that humans are still required to do science is because the computer is only as good as the data set that you train it on or the input. And so I'll talk a little bit about how we read the single cell RNA-seq data and just point out a couple places where it's like, you have to be really careful because, if you pick your own story. So basically like the bottom line the goal of single cell RNA sequencing or the analyses that we use and the way we generate the graphs is to take a highly dimensional data set. And that means, we have like thousands of genes and thousands of cells, and each of those are their own dimension. How do we even wrap our heads around that? Like, how can we represent it in a way that we can consume and think about? And so, you might have heard the acronym TSNE, or here they use UMAP, which is a better version of TSNE. And they're taking this highly dimensional set and they're reducing it to something that has very few dimensions. And this is similar in a way to a principal component analysis. But what principal component analysis allow you to do is to say, like, here are two components of the data, like maybe it's brain region and genotype. And these are how the different groups of cells cluster depending on these two variables. But single RNA sequencing doesn't really allow you to do that. So I'll just point that out, that there are a couple of things. If you look at the graphs, you're like, okay, well, this group goes here and this group goes here. Therefore, they must be really different from each other. Or like this group is really tightly packed and this group is much larger. And so, it means that in this group, there's a lot more like homogeneity, whereas in this group, there's a lot of heterogeneity. You can't make those kinds of conclusions. So because, this is not an episode of like single cell RNA sequencing 101, or, 0.5, because I know I'm not an expert at all. I do have a link that we can put out there that I think is very interesting. But one of the places that really depends on your input and can change completely how the results come out is that, so UMAP takes all of the individual, like those dimensions, each individual cell at each individual gene and its expression levels, and it determines which other data points are closest to it and the likelihood that those two data points are going to be connected. And it does this by sending out has these a radius going from one cell or from one data point to every data point within a certain a distance away from it. And you set that radius. So if you set it really short, you may have a population of cells that's this big, but now you're clustering it and you're saying like, here's this population of cells and all the other ones are different, when the science gods know really that it's just one big population. If you set the radius too large, then you're going to be pulling together cells and saying like, these guys are all in the same population when they're really not. And the thing is, like, we can't know if they are or if they aren't, because we are not the science gods. So we just have to use judgment, our, human judgment, which is questionable, even among us scientists, which is
that's what we have, right?
It's all we have. Yeah. And I think the other thing to bring up is that it really that judgment depends on what your question is. So for example, I have a student who's working with me who's wondering why we should write. We're looking at differences between the subventricular zone and how it compares to the cortex in bulk RNA sequencing. And he had written, the title is Cortex versus SBZ. And we were trying to explain to him that implies that our question is, how does the cortical transcriptome change relative to the subventricular zone? When actually our question is the reverse. And to try to, it seems very like semantic and like, wow, who cares? But like those are meaningful when it comes down to interpreting differences between groups and which direction things go. Like I mentioned earlier, some genes, it's not that they go down, but they're actually going down in the conditions where microglia are not present. It's really just that they fail to rise. So these differences are meaningful. And so I think you just have to read the results carefully and think about like, okay, well, what is that? What are they able to say and what are they not able to say?
So isn't the goal to try and find genes that are key for recovering from SLI with the goal of eventually being able to tweak those in people? But the problem is that you get a whole bunch of metabolic genes, right? Which you need to make more protein, and that's not useful, right?
Yeah, exactly. You see things like let's see, where did I write it here? And I kind of laughed because I was like, I mean, that's like, so you mean like it just, that's stuff that cells need to do to survive.
Yeah, I mean, if you knocked out protein translation, you'd interfere with spinal cord injury recovery, but that is not a viable approach, right? You need something really specific, and there's so many genes that it's hard to tease it out, because what you have to do eventually is to go in and test some of them, right?
Exactly. So let's see. There's a whole lot of stuff that the going up and going down, and ultimately the message is that this.
That's the name of this episode: going up and going down.
Yeah. I hope you don't have motion sickness. If you do, don't listen to this article or listen to this episode. But essentially, they're able to show that without that the point I want to make, the overall like take home message of this is that, these little cells, these microglia, which we have unfortunately tended to think of as hindrances rather than allies in many situations, that they have major roles in regulating like the cells that do a lot of the heavy lifting in these scenarios, like the macrophages and the microglia. Everybody's talking about the glial scar and what's happening with those and like infiltration of immune cells. And it's like, well, like, let's take a step back. Like, who's orchestrating all of that? And this paper is suggesting that one component of that regulation and the, control or, determining the extent of recovery is microglia or microglia. I don't know. This is a word. It's like, it's supposed to be plural, but you say a microglia. So it's very confusing. It just feels wrong every time I say it. But, so I think, I think those are the take home message. That's the take home message of
Yes, microglia, the kind of like the resident sentinel of a particular tissue, so that they, if something goes wrong, they are the one that waving, the flag around saying, "Please come!" Like something is wrong here.
Yeah.
Okay.
I mean,
like, are they the? So you mentioned that they can do some. They can phagocytose and eat debris around them and then display them again. Are they the only cells that can do that in the spinal cord when injury, like before the injury really kicks, like at injury time? Are they the only?
Oh, at injury time? You mean, or like prior to injury? Or does anybody
or prior
else
to injury? Like, are they the one that is responsible for detecting the injury? It seems like that is the case from this paper.
Yeah. So it's complicated. It depends.
Okay. Oh, no.
But I think if you were to define the what you're essentially calling, what you're talking about as antigen presenting cells,
right?
If microglia are, generally speaking, like they're the antigen presenting cells, but I would never in 100 years say that they're the only ones that express the machinery that's involved in presenting stuff.
Okay.
I think there's a lot of plasticity. I mean, this is a really important organ we're talking about here. And so I think that, and you can have, for example, they talk about in this paper that in the app that there's a specific, like I said, it's admirable that they're like, hey, look at this specific receptor ligand interaction that's, between two genes that you've never heard of. But that's important. If you knock out or if you deplete the microglia, this interaction still happens, but instead of being paracrine or intercellular, it becomes autocrine, where the astrocyte will produce its own ligand. And, so like there's a quite a bit of, kind of backup plans. So that if something were to break down in one part of the response, there would be a way to come back from that. So I think
in the end, I mean, most of what we do is just looking at how systems compensate for the loss or gain of something. And it's very difficult to say this is the causal role because there's so many things in between that can happen that compensate.
Right. I once had a professor say to me that he was like, arguably, if you see no difference, it actually means it's really important. I mean, he was just saying that to make me feel better,
I'm doing really important science 24/7.
Well, I mean, but then it's up to you to figure out why, I mean, or you could just be like, the NIH is not going to pay me to study this, so I'm going to go study something else.
So what Jason just said is important because you have, in the discussion, they point out, actually in the results, they point out all these little networks and ligand receptor interactions. And you can imagine people going through now and knocking out each one, and they may not find out anything because it's a big, complicated network of things. It's not one thing it's off, or not one thing that you need. So I'm not sure where you go with this, because you can imagine wasting a lot of people going, okay, you knock out this gene, you knock out that gene, and on their own, they're not going to tell you anything, or they might not.
Yeah,
could be wrong.
They can try to knock out something that controls several genes at the same time. Perhaps,
right.
If they can identify it, which is weird because they did identify something that you talked about earlier, which is CCL2 and TLR2.
Yeah.
But then during the single cell sequencing, they don't talk very much about that, which is a bit
that's
odd.
true. And also, let's see, it was through bulk sequencing that they found that. Because sometimes, like, for example, you'll see that, like, the RNA is present, the RNA for a target is present in a population of cells, but you can't detect the protein. Like, that's the central dogma is that, like, DNA leads to mRNA, leads to protein, and there's so much more in between that.
So like on a single cell level, it's just so tight, it just gets so diluted by the other genes. Yeah.
Possibly, or you need to have a constantly available pool of the transcript, but you may not want the protein around. So that at a moment's notice, the complex that's involved in degrading the mRNA can be shut down, and you can have that you have reduced turnover of the transcript, And that allows you to generate a lot of protein very fast. So it's all complicated and nobody knows anything. And
So I have a broader question, which is, in these experiments, so they do a spinal cord injury and the mice recover eventually. So, but I would argue that's not what we're interested in, because some people can recover. But what about the really traumatic injuries where it's hard to recover? Maybe they should see in their mouse model what's involved with that, and what they could add back from some of these networks that might improve that, right?
Yeah, that would be a good approach. Definitely looking at severity of the injury and how that's going to affect the increases or decreases or timing. Yeah, that would be very interesting, because in both, that's very relevant. And in both cases, you know that something has changed. You just have to, it's about more about extent rather than it is about like yes or no.
Because maybe this study identifies some fundamental pathways, and then you say, okay, if we do a really irreparable injury, then what of those has trouble or something like that? You know?
Yeah. That's definitely more forward focused.
All right. Thank you, Vivian.
Cool.
My pleasure.
That's very cool. I'm a little upset because I don't have an answer.
I mean, welcome to my life. So I don't know if I know that, you know, we're coming up on ending time, but I do just want to share why I say that it's my life. During my dissertation, I worked on a protein whose an enzyme whose product you can't detect unless you are a liquid chromatography mass spec wizard. So I had no way of seeing its levels. And also it's one enzyme in a group of several enzymes. It is like the least expressed postnatally. And it's highly expressed in a population of cells that was not impacted by I won't go into the details, but basically I spent a lot of time thinking about and explaining how you have these very complex interactions that don't make any sense. Like reviewers are asking, like, why would RALDH2 be important if right next door RALDH3 is like, you know, just super bright, you know, and RALDH2 is like barely detectable. And, you know, it gets down to things like, well, the retinoic acid that RALDH2 generates goes to a specific little pocket in the cell, which is important for. And so I swore I am never going to study a protein that's so lowly expressed ever again. Like I'm only going to study GFAP or something. And guess what I worked on? What I'm working on for my postdoc? A protein that is barely detectable in microglia, but it's just gangbusters in blood vessels. And this is the same thing for my dissertation too. It was like super high in blood vessels, and then like not detectable anywhere else. The project I'm working on now I'm going to be going into is like, again, it's an enzyme that's in a class of 3 enzymes. The expression patterns of which are really unclear and nobody agrees on, and also you can't detect its nitric oxide. You can't detect it unless you are, you know, mass spec wizard, or you can only look at it indirectly. It's like Schrodinger's cat, you know, or like electrons. If you look at it, it's going to be somewhere else, you know. So it's my life, FML.
That's your life, and this is TWiN 34. And there are show notes. They're at microbe.tv/TWiN. You can send your questions and comments to twin@microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Our presenter today, Vivian Morrison, is at Tulane University. Thank you, Vivian.
Yeah, it was fun. Thanks, guys.
Jason Shepard is at the University of Utah. He's Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Vivian. Thanks, Vincent.
Tim Chung is at New York University. Thank you, Tim.
Thanks, Vincent. Thanks, Vivian. And I'd like to hear more about nitric oxide at some point. I think it's a under-recognized under talked about neurotransmitter, if I remember correctly.
See, I'm not talking about it in neurons. Okay. Not yet, anyway.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.